Okay, I think we should get start going. Today we have with us Dr. Aitana Sogorb. She's originally from Alicante, right? And she's a senior research fellow at UCL, University College London, and beneficiary of a prestigious race against, it's a prestigious award, right? Of a race against dementia fellowship, which is basically a junior research group leader fellowship in the UK. And she's a biologist by background and specialized in, in biomarker research and laboratory research, and specifically biomarker research of dementia biomarkers. I think also Alzheimer's disease, but now in her own group at UCL, she focuses more on biomarkers for frontotemporal dementia. And looking forward to your presentation.
Thank you very much. Well, thank you, everyone. Thank you, Michelle, for inviting me and giving me the opportunity to talk today. So, so I'm Aitana, and today I'm gonna kind of summarize the work that I've been doing, like with my team in the past few years. But first of all, in the department, when we give these seminars, it's usually internal speakers, and we have this journey to the Dementia Research Center. So I thought it would be nice to have that today as well. So I kind of try to plot here my journey to the point where I'm now. As Michelle said, I'm from Alicante, and I studied biology there at the Universidad de Alicante. And I did licenciatura. Back then was 5 years of university degree. And on my 4th year, I went to Barcelona. I wanted to be a marine biologist, actually, but then I went to Barcelona and I took a lot of subjects that were not available in Alicante, and then I fell in love with neuroscience. And when I came back, I did a summer internship in El Instituto de Neurociencias in Alicante, and then I decided to do my master's and my PhD there as well. And during my PhD, I also did an Erasmus Plus fellowship and internship in Sweden. So I worked in the laboratory of Kai Blennon for 4 months. And I kind of, my PhD was all about fluid biomarkers in Alzheimer's disease. I was doing Western blot mainly techniques, and the internship in Sweden kind of introduced me to the world of ELISAs and other immunoassays. And during my PhD, I was working on presenilin-1 complexes in spinal fluid from Alzheimer's disease, but also Down syndrome. So yes, when I finished my PhD in 2018, I moved to the UK. And I started as a postdoc in the UK Dementia Research Institute. And I was between Henrik Zetterberg and Bart de Strooper groups. I was actually hired by Bart de Strooper. And then I'm saying that I was with the University of Gothenburg and the KU Leuven because I travel a lot. So I was between the 3 countries, going to Leuven to learn some things, going to Gothenburg, and so on. And then after 1 year and a half, I applied to a second postdoc, still at the University College London at the UK DRI, and then I started working in frontotemporal dementia, because before I was working on Alzheimer's disease mainly. On my first postdoc was on microglial activation markers, mostly in iPS cells, but also mouse models. So then when I was my second postdoc, I applied for this Race Against Dementia fellowship that Michelle mentioned. And this is basically a fellowship available in the UK for 5 years, and is funded by a Formula 1 driver. And it's very curious because they want to translate the philosophy of Formula 1 to dementia research, which is a very interesting approach. But, you know, it's a very good pot of funding, and we have very fun things as visiting factories, going to Grand Prix and things like that. So it's not all science. And then when I was doing, like in my first year of my fellowship, I then applied for a promotion at UCL. So then I became a senior research fellow at UCL, which is kind of a next step. And that is where I am right now. So today I'm going to talk about the work that I've been doing on my postdoc, basically, since I joined UCL. And I know in this room, the audience is very aware of this, but just to set everyone on the same page, I work mainly now on frontotemporal dementia, and FTD is a very complex and heterogeneous disorder. So the clinical manifestations are very different and various. It can be affected the language, the movement, or the behavior. And then we have a different underlying pathology as well. And the relationship between the underlying pathology and the clinical manifestations is not linear. And on top of this, we have an extra layer of complexity, which is genetics. Up to 50% of cases of FTD are caused by a genetic mutation. The main mutations are progranulin, C9, operating difference 72 expansions, or tau. And in this case, and I'm going to make a stop here, and I'm going to ask you to hold that thought, the relationship between genetics and underlying pathology is more linear. And this is something that is going to be important later in the talk. So as we know, the clinical disease appears very late on the stages, and we have changes very early before the symptoms start, and changes in CSF and serum occur at very, very early stages. So this is very important because for frontotemporal dementia, we still don't have any good biomarkers for diagnosis, and therefore the development of therapeutical interventions is very challenging as well. So we need to kind of address this problem and solve this problem. So the questions that we wanted to address in the research is, first, can we detect neurodegeneration in FTD? Are the current measures for neurodegeneration good for FTD? Then can we assess cellular dysfunction with fluid biomarkers? And finally, what is left to learn from fluid biomarkers in frontotemporal dementia? So the approach, and I'm going to come back to the first slide, that the research group that I joined at UCL had to solve these questions were like, okay, if we know that the genetics have a more linear relationship to the underlying pathology, if we study these fluid biomarkers in genetic frontotemporal dementia, we have at least a little bit more of information, and we can maybe better understand what the fluid biomarkers are telling us from what is happening in the brain. So that is one of the goals of this cohort, which is the Genetic FTD Initiative, and for short, GEMFI. And GEMFI is a multi-center cohort study with 49 centers now across Europe and Canada. In Spain, you have in Hospital Clínic Barcelona, Sant Pau, Santander, and Donosti. And basically, it has been running for, I think, around 13 years now. So it's a longitudinal study. And in GEMFI, there is a recruitment of mutation carriers of the mutations causing frontotemporal dementia at both presymptomatic and symptomatic stages. So basically the way GEMFI works is that when there is a memory clinic and there is a patient that is suspected to have FTD, they go to genetic screening. If they are positive for one of these mutations, all the family members are asked to be part of the study. Some of the family members will be carriers, but they don't have symptoms yet. They will be characterized as presymptomatic or put in the presymptomatic group, and other family members will not be carriers because we also have not the children of the person, but also the partners or the carers. And they are also asked to participate in the study, and they are the non-carriers or healthy controls. And the good thing about GEMFI is that it has been running for a few years. It's longitudinal, as I said, and we have unified and standardized procedures and protocols, SOPs, in all the centers. So the data is collected in the same way, the samples are collected in the same way, the cognitive assessments are done somehow in the same way that they can be unified. And one of the students, one of the PhD students in the London group, they created also an iPad app, which is called Ignite, that is now implemented in most of the centers for also a cognitive assessment. So the creator of GEMFI is Professor Jonathan Rohrer, who is my line manager or PI, so I'm kind of somehow associated to his group in London. So therefore, in London, we are the headquarters on GEMFI, and I'm so lucky of that because we hold 50% of all the samples in London. So I have a very easy access to all the samples from the participants. And I think to this point, the numbers are around 1,600 participants recruited and follow up during years. So the number of samples that we have is even more than this. So just So it's clear because the results that I'm going to show today are all based in GEMFI, and even though they are not exactly the same cohort of participants, it's very similar because we try to optimize the sample, and most of them are matching the same participant, and sometimes we we increase the number of samples, or if we don't have samples, it's a bit less. But in general, we classified, as I said, our participants in non-carriers, presymptomatic, mutation carriers, and symptomatic. And actually, the icons here, I chose them intentionally. In GEMFI, all the symptomatic mutation carriers are older than the presymptomatic. So that is something that is very important to take into account when we analyze our data, because there might be some changes in biomarkers that are more related to aging than to the actual condition. And in the control group, we have both, but the older group is less common than the younger group. So controls tend to be younger as well than symptomatic and somehow match aging with the presymptomatic. So there are two ways in which we approach the data. We kind of can do presymptomatic, symptomatic, and non-carriers all together, or we can divide them by genes as well. So if we look at the mutations, we have symptomatic C9, progranulin, and MAPT, and also the sizes here are intentional because the MAPT mutations are rarer, therefore we have very few participants who have a MAPT mutation, but we have quite a lot of C9 because it's more common. The same for presymptomatic, and then the non-carriers, they are not carriers of any mutations. So even though they come from a family of C9 family or MAPT family, we group them all together. So this is in general how it works, and how I basically, most of the results, I will present them in this kind of format. So you just have an idea of the pros and the cons of the cohort. So I'm going to start by the markers of neurodegeneration and how we have been studying neurodegeneration in GEMFI and in FTD. And as most people in the biomarker field, we have been studying neurofilament light, and we use the CIMOA because we are based in London within the Biomarker Factory, who is led by Henrik Settenberg, and therefore we have access to 4 CIMOAs, 1 Alamar, and different machines. So we use CIMOA for measuring neurofilament light in plasma. I think the next slide is serum, but it says plasma. But this was in plasma, and this was the work done by one of the PhD students, Caroline Heller, and she has been studying the levels of neurofilament light, and we can see that, as expected, we have an increase in symptomatic mutation carriers, but actually we always find this kind of high variability in the progranulin mutation carriers. And then we did a collaboration with the twin cohort in the US, which is called ALL-FTD. So it's very similar, the structure, as GEMFI, but it's based in the US, and we collaborate quite a lot with them. And so Julio Rojas was the author of this paper, in which, together with Caroline, they unified and then put together all the NFL data available for both GEMFI and ALL-FTD, and they looked at the levels according to the clinical syndrome of the patients. And we see that NFL is increased, but also with high variability in most of the cases. So the other thing that we've been doing in GEMFI as well, this is a paper from the group in Rotterdam. They also took the NFL levels, but this time in serum, and they looked at the NFL levels in a longitudinal way. So basically, they calculated the rate of change in these participants in terms of cognitive decline, and they correlated it, or studied how the levels of neurofilament light will correlate to this. And what we see is that when the symptom onset is close, the levels of neurofilament light increase. And this is the group that we call here converters. So next, can we assess cellular dysfunction with fluid biomarkers? And we have been working in different aspects of cellular dysfunction: neuroinflammation, lysosomal dysfunction, and synaptic dysfunction. And I'm going to show most of them, if not all of them, unpublished results from the group in terms of each of these cases. So first, in terms of neuroinflammation, I know it's neurodegeneration, but we also, I wanted to include GFAP, and this is something that Caroline is doing, finishing her thesis, and this is unpublished. And Caroline did the same study that Rotterdam group did with NFL, but with GFAP levels, also measured in plasma with the Simoa assay. And then Caroline divided in the kind of the asymptomatic or the presymptomatic in both asymptomatic and prodromal, and we can see how the trajectories of of GFAP are very similar to NFL. They kind of increase when the symptom onset is close, and this is kind of similar analysis with the annualized rate of change, which is statistically significantly higher in the converters. So the next thing is inflammatory markers. And this is the work that one of my research assistants, Katie, have been doing for the past year and a half. And for inflammation, we've looked at we use this company, which is called Olink. But back then, when we started this study, we didn't have Olink at UCL, but now we have an Olink machine at UCL. So we sent the samples to the company, basically. And Olink is, for those who are not familiar with this technology, is called proximity extension assay. And it's basically an immunoassay as a conventional ELISA, but to the antibodies, you have a DNA proof that will pair to another DNA proof, and then you will do a PCR. So this technology is very, very sensitive because you amplify your detection, your signal with a PCR. And actually, Olink only needs 1 microliter of sample to measure, in this case, is 92 inflammatory markers. It's the panel that they call target 96, but I think there was 92 proteins. So we did this in CSF samples from Genfi. And this is a bit preliminary data. We need to refine a little bit more the analysis, but we basically studied which were the proteomic inflammatory profiles in each of the genetic forms versus controls, but also presymptomatic, symptomatic versus presymptomatic mutation carriers as well. And we see some interesting results here, as it's, I know it's very small, but you have to believe me, MMP10 here keeps appearing on MAPT mutation carriers. So it's something that it seems to be in MAPT, but not appearing in the rest. And these studies are very useful because they gave us a lot of information, but also they gave us somehow hints on, okay, what should we be looking at? What can be a possible biomarker of neuroinflammation? So yes, it's actually very, very important, but still is a work that it's a work in progress, and we need to refine a little bit more the analysis and the thresholds for this. And a part of Olink, Katie also has been working on microglial activation markers, and we've measured in the same cohort as Olink, the same samples, CSF samples, both YKL40, YKL39, and also K3 oxidase. For YKL40, we used a mesoscale discovery platform, which is ELISA activated with electricity instead of a colorimetric change, and it's meant to be more sensitive than conventional ELISAs. So we saw that the levels of YKL40 were significantly increased in all symptomatic groups, but again, we have a high variability in inflammatory markers in each of the groups, and mostly in the progranulin group. We always have this high spread that we actually want to further investigate what is going on here, because we keep seeing it in all the studies. We also found differences in YKL39, but only in C9 and progranulin, but not in MAPT, and we were not able to find any differences in K2 oxidase. And these two analyses were studied or were measured using conventional ELISAs, commercially available ELISAs, which we don't know. They are meant to be validated. We needed to do a bit more of validation. So it would be ideal if we could have all of them in the same platform. So another project coming from here will be to maybe try to adapt the mesoscale discovery and develop assays for these two analyses, so we can have a panel in the same plate with all three of them and see if it works. So the next thing is lysosomal dysfunction, and this is quite interesting in FTD and, well, in genetic frontotemporal dementia, because one of the mutations causing FTD, progranulin, the progranulin protein has a lot of functions in the endolysosomal system. So we were actually expecting to see a lot of changes in endolysosomal proteins in progranulin mutation carriers. And we decided to measure this panel, which is a targeted mass spectrometry panel in collaboration with the University of Gothenburg, that they developed from a broad proteomic study. So the way they developed these targeted mass spectrometry panels is basically they did, in Alzheimer's disease, they did an unbiased proteomic study. And from there, they have been picking up hits, and also together with the literature, they have been creating these panels. So just so you're aware, this panel was meant to be very profiled to Alzheimer's disease, but we thought that will be interesting to see what's going on in FTD and mostly in progranulin. To our surprise, we didn't find any changes in the progranulin mutation carriers, which are the blue shades in here. But what we found that for a few proteins, DPP2, cathepsin D, and XB, there were significantly changed in either presymptomatic or symptomatic MAPT mutation carriers, which was very surprising. Actually, at the beginning, when we saw this result, we thought, okay, we don't know what's going on here. We really don't know the story behind this. We still don't know it, but just hold this in your minds, because it will be important later as well. So the next thing is the synaptic dysfunction, and this is something that we published a couple of years ago. We also used a targeted mass spectrometry panel of synaptic proteins in collaboration with the University of Gothenburg. You will see that most of the things that I do is in collaboration with Sweden, because it's kind of my second main institution, so I spend quite a lot of time in Sweden, going there, preparing all the samples, running all these experiments. So Johanna and I spent quite a lot of time together doing this targeted mass spectrometry panel of synaptic proteins in FTD. This panel was actually developed the same way as the lysosomal one. It was from an Alzheimer's disease study together with literature. So it was somehow more profiled to Alzheimer's disease. And what we actually found when we measured the CSF from GenFi was that most of the proteins of the panel were specifically increased in symptomatic MAPT mutation carriers, but not in the other groups. Except of the neuronal pentraxins that had been reported before to be very important in neurodegenerative diseases. They are not specific of FTD, but the changes are quite pronounced in FTD, and they are decreasing all symptomatic mutation carriers when compared to their presymptomatic groups and controls. So after seeing these results, we thought, okay, I also presented these results a few years ago in an internal GenFi meeting, and someone asked me, where are all these MAPT mutation carriers coming from? Are they from the same center? Because as I mentioned before, we don't have that many MAPT mutation carriers. So in the end, we end up with maybe 12, 15 individuals in this group, but they come from different centers. I checked this. So we really don't think this is something related to the sample or to families or something like that. Might be because the panel was developed for a profile for Alzheimer's disease, but I'm not sure about that explanation, because Alzheimer's disease is a secondary tauopathy, FTD is not. So what is going on here? So also this was kind of the pilot data and the basis of my fellowship application, because my project focuses on mainly measuring synaptic dysfunction. And this is also the basis of a proteomic study that I'm going to show now. In the next step, what is left to learn from FTD fluid biomarkers in FTD? Mostly everything. We don't know that much, so there is still a lot that we can learn from there, and a lot of things that we can still do in this. And this is what we started to do. So based on the results that we saw in the synaptic panel, we said, okay, something is different here, and we see synaptic dysfunction in frontotemporal dementia as well. But why, if we are not looking at the right actors here, we are not looking at the right proteins, so what's going on? So then again, spending quite a lot of time in Sweden with Joel and Sophia, we did this unbiased and not unbiased and targeted proteomic study in GEMFI, and we included 238 CSF samples. And this is now pending of a couple of authors to approve the conflict of interest statement to be accepted in Science Translational Medicine. So we are very happy about this. And basically for this, we use a mass spectrometry approach, which is called tandem mass stack proteomic, in which you label with these stacks each of your samples. So each of your sample has a different label, and then you can pull them together and analyze them together in the mass spectrometer. When we did this 1 year and a half ago, the company only had up to 16 plex. I think now you can multiplex up to 31 different samples. So then we labeled all the samples. It took a few days working very long hours, and then we analyzed them in the mass spectrometer. And the approach that the TMT and the type of analysis that we did in the mass spectrometer is called data-dependent acquisition. So there are two kind of variables, two ways you can acquire the data in mass spectrometry: data-independent acquisition or data-dependent. And the difference is that in this data-dependent acquisition that we use, you are on the risk of losing low abundant proteins, because what it does basically is the mass spectrometer selects the most abundant peaks, and then it amplifies that and it measures that. So here we're at a risk of losing low abundant proteins, but we wanted to have an information of mostly what was going on, like a big picture of everything. Maybe if you are willing to find, for example, TDP43 cryptic exons or TDP43 fragments, it will be better if you use the other approach. But for us, it was more to have a sense of what's going on here. And then we did quite a lot of analysis on the data. We managed to measure in on the samples included, after outlier removal and contaminant removal, 1,992 proteins in all of them. So you imagine the amount of data that we had that for 238 participants. So we first looked at differential protein abundance and the protein profiles in symptomatic mutation carriers for each genetic form versus control. Then we looked at the overlap and differences of the 3 genetic forms, but also which changes might be more specific of frontotemporal dementia and to each of the mutations. Then we wanted to look at the pathways. So we wanted to make a sense of all the data and see if we could understand what was all these changes in proteins telling us. And finally, we associate these changes to clinical parameters that we have in GEMFI, because as I mentioned before, for GEMFI, we collect brain volumes, brain imaging, cognitive scores, and we keep that for all the participants, and we have a big database of that. So then it's very easy for us to correlate the biomarker data to that. So I'm going to go now through all the figures of the paper, basically, and this is the first view, which is the proteomic signature, and I acknowledge that this is very small to see in the volcano plots, but I think this is quite a nice summary of the volcano plots. So basically, to create this heat map, we took the 25 top hits of each of the genetic forms, and then we put them together. So in total, there are 62 proteins, because most of the top hits were common in the 3 genetic forms, so we didn't want to repeat them. So we see actually very common proteins increase, which are actually the drivers of the changes, which are neurofilaments, YCL40 as well. We also have fatty acid binding protein 3 and 1433 proteins, as well as UCHL1. And then on the bottom here, we have a common decrease in neuronal pentraxin families. But the other things that we observe here is maybe the shades are not very clear and you can see it, but we saw proteins like PLBD2 and DNase2 that had different direction of changes in the genes, and actually then they will appear back, and in an analysis that we didn't include it in the paper, they actually came as the best predictors or the best proteins differentiating MAPT from the others, because basically in the case of PLBD2 is decreased in MAPT but increased in C9, and in the case of DNase2, I lost it now, here, is decrease in MAPT, but increase in progranulin, and no change in C9. So there were some interesting things that we can take from here. And the next thing that we did, and actually this was all colored with the different colors here, so it was clear to see, but I don't know the format of the PowerPoint, then it changes. It always happens to me. But basically, we wanted to know first, okay, from all the changes that we see in GEMFI, what is really specific of frontotemporal dementia? So we were so lucky that we had also data available from the EMIF cohort, because actually the study was done in Gothenburg as well. And then this paper was published as well using tandem mass stack proteomics. I think this was in ADNI, but I'm not sure right now. And the database was available. So we basically compare our data set with the other two to see, okay, the changes that we find here, are they specific of frontotemporal dementia, or they are common neurodegenerative diseases markers? And actually, from all the proteins, 1,192 were common in the other data sets, and only 221 one specific of GEMFI, but still is something. And then in the same way, we also explore the overlap between the three genetic forms. So we actually have 149 proteins that are only changed in our study in C9. Then maybe if we do another study with another cohort, that might change. 26 in progranulin mutation carriers, all symptomatic, and 14 in MAPT. And then also very interestingly, because we explore the overlap between the three groups, we have 43 proteins that are common between C9 and granulin, but not in MAPT. And actually C9 and progranulin mutation carriers, they shared an underlying TDP43 proteinopathy. So maybe if we then, the next thing will be to pull out these lists and study a little bit more these lists to see if there are some markers that could be related to a TDP43 proteinopathy, and in which way they are related to that. So the next thing we wanted to study was, okay, are these changes present at earlier stages of the disease? So what we did here was to study the proteomic changes that were related to the mutation, and they were somehow also present at presymptomatic stages. And for that, we ran linear models for the protein here with the affected condition as symptomatic or presymptomatic as a categorical variable of the model. And what we saw is that for C9, open reading frame expansion carriers, calretinin seems to be associated and also changed at presymptomatic stages of the disease, as well as GP1 here, which is not statistically significant, but it shows a trend. For progranulin, obviously we had granulin, but then we also have NAGA and RNF13 that were changed and differently changed from the other groups. And for MAPT, we got PA15 and semaphorin 6 that showed changes that were different from the other groups as well, statistically significant. So here we have some proteins that might be somehow related to the genetic mutation, and they are also starting to change at presymptomatic stages. So that will be indeed very useful. So then we we did this pathway analysis, and from this we did also a WGCNA approach, and our analysis generated 12 clusters. Here are only the 6 most interesting ones. And then after this WGCNA, we apply a gene ontology approach to somehow name those clusters, to basically know which were the top proteins, which was the role of the top proteins driving the clusters. So this is the way the clusters are named, but it's not a very strict name, but we wanted to somehow identify what was going on with the clusters, basically. And we had these core markers, which contain neurofilaments, 1433 proteins, YGL40, which we were actually expecting to be changed, and they actually changed together, and they are significantly increased in all groups. Then we also had, interestingly, the synapse cluster, which shows a decrease in all symptomatic mutation carriers when compared to presymptomatic and to controls, which is completely the opposite that we found in the synaptic panel, that most of the proteins included in the synaptic panel were specifically increased in MAPT. So there is a lot that I can study here now. And then very interestingly, and coming back to the results that I presented before, we were very shocked when we saw this cluster that we named lysosome cluster. And it was only and specifically significantly decreased in symptomatic MAPT mutation carriers, but not in the others. And actually, if you look at the top drivers, here we have DNase2 and PLBD2. So these proteins, again, they come up in the analysis. So it will be interesting to study this as well. And then we also see this cluster of immune response that shows a small trend, but is not significant. So then with this, we wanted to see, and we correlated the clusters obtained with the clinical parameters as in brain volumes and also CDR scores. And what we saw that those clusters that were increased, they were obviously negatively correlated with the loss with the brain volume and with plasma NFL, and the increase of cognitive scores, they were positively correlated, and the synapse and neuronal development clusters, they were correlated in the other way. So the lower levels of these proteins, they are correlated with higher levels of these, and the other way around for this. And then we saw a lot of, not that much correlation when we looked at the only symptomatic carriers alone, but then correlations in the presymptomatic mutation carriers. So this is something very interesting, and that we want to pursue the next step or the next chapter of this study. And the last thing we did was this analysis, because one of the reviewers, they actually asked if we could do the same in a sporadic FTD cohort and in a longitudinal cohort. So this was obviously impossible for us because we didn't have enough samples for that. But in GEMFI, even though we don't collect CSF samples in each of the visits, we do have a cognitive score. So what we did was to calculate a rate of change or a kind of a change on the CDR sum of boxes score, and then we correlated to the baseline levels of these clusters, and we saw that lower levels of the synapse cluster or neuronal development cluster, as well as protein processing, they predict higher scores, higher cognitive scores, and then higher levels of core markers, actin binding, GAG processing, they predicted higher scores as well. Lysosome, the same, lower, but I think this is mostly driven by MAPT and immune response as well. They were predicting higher scores of the cognitive, the cognitive assessment. So yeah, this is in a nutshell all the work that we've been doing in a couple of years. And then the last thing that I wanted touch upon is extracellular vesicles, because I'm now working on extracellular vesicles. And the results that I'm going to show today are the work from my postdoc, Luis, and my research assistant, Ellie. Luis has been with me for 1.5 years now, and Ellie started just at the beginning of the summer. And they've been working on characterizing the extraction methodology for extracellular vesicles. And this is just an example on some markers that we've done, Luis has done in FTD, sporadic FTD versus the controls. And he has done some characterization. And we actually found that in sporadic FTD, the number of vesicles is significantly lower than in controls. So this is actually something to take into account when we measure biomarkers in extracellular vesicles. We will have to normalize to the number of vesicles and the concentration of vesicles that we have in the end. This is just some very nice pictures of EVs. And Luis, he's a cross-center postdoc, which is between UCL and Cardiff University. And in Cardiff University, he is based in an immunology that now is part of Dementia Research Institute groups, but they have the PI has a strong basis on immunology. So they are working a lot on the complement system. And that's what Luis' project is about, to measure complement proteins in extracellular vesicles in FTD. And this is data from plasma from this sporadic FTD cohort that we managed to send him. These are all the analytes in the complement cascade that they are actually measuring. They measure a lot of activation factors. And we saw some changes in factor B, C4, and IC3B. Not that significant here. And then he tried to do the same with the extracellular vesicles. And it looks very kind of shiny, but actually this is not normalized to the amount of vesicles, the concentration of the particles. So when he looked at extracellular vesicles, there were more significant things, factor B, factor H, propyridine, and C4 as well, that were quite promising, C5, C7, but we still need to kind of normalize this data and see if they are still significant, which most of them might not be. So I think I'm going to stop here, and I just put together like this broad summary of what I showed today, that NFL and GFAP levels increase towards symptom onset in genetic FTD. We see different inflammatory profiles in the genetic forms, and also some changes between presymptomatic and symptomatic mutation carriers. We see something unexpected, which is somehow something is going on the endolysosomal system in MAPT mutation carriers that we still don't understand. And this was actually one of the bases of the grant that I got to hire Ellie. So she's going to be working on extracellular vesicles, but lysosomal proteins in them. We identified specific proteomic profiles in genetic frontotemporal dementia, and we see some changes, some proteins that change at earlier stages. And finally, for now, we know that there are lower levels of particles in FTD, but this is a lot of work in progress, so a lot more things, exciting things will be coming. So yeah, this is me. I would like to thank all the GEMFI participants, because and the carers, because with the samples from the participants and the willingness to participate in the study, we actually couldn't do any of this. And then all my collaborators and my team, and thank you very much.
Yeah, thank you, Aitana. Do we have questions from the land? No. I'm sure, yeah. Yeah, I will come back to you later. So what do you think about these recent developments for extracellular vesicle, plasma extracellular vesicle, tau and TDP-43 measurements, and like separating 3R tau and 4R tau? Because if I remember correctly, in this paper, in this Nature Medicine paper, they did have behavioral variant frontotemporal dementia, sporadic forms, where they expected by pathology, they expected like about 50% to be TDP-43 positive and the others tau positive, which is what they found, but they don't have any ground truth actually.
Yeah.
For the PSP sample, they obviously, they knew what they expected, and that played out quite nice, but actually like these C9 mutations or progranulin mutations, that would be really nice for like more specifically testing without having pathologic information, the specificity of these markers for TDP-43 actually, right?
Yes, so. Yeah, definitely. Okay, so you put me in a tough spot here. I'm gonna be honest with you. I don't know who is listening online, but I was very excited about this work, and I see two main kind of, I will love to see a replication of it, just because there is a lot of controversy. I've been in the work of extracellular vesicles not that long, like a couple of years since I was awarded the fellowship. And what I've seen is that there's nothing, there is not a consensus of anything, not the extraction method, not which markers of neuronal specificity to use. So yeah, it looks very good, but I really don't know if what is they are actually measuring there, if it's really neuronal derived extracellular vesicles, because I think they did not doing immunoprecipitation with L1CAM and doing immunoprecipitation. I think they have both.
I'm not sure about that either, but I think as I read the paper, they were really careful about not saying neuronal
derived. Yeah, but I think they did also immunoprecipitation with L1CAM. But yeah, the other thing is that, to my experience, we haven't been able to work the TDP-43 CMO assay in any of our samples. it doesn't work. So I was really very surprised that it worked in this. So actually it will be great to see a little bit more like a replication of it as well. The other thing that I think it's a definitely very promising study and something that we want to do now. We have now in place one soon to come, the second one. We have an Alamar and Argo in the system. So this is a new technology which is called Nulisa technology, and it's meant to be 10 times more sensitive than Simoa. And they do have TDP43 in the panel, in the CNS panel. So I would be very excited to see, and actually I've already asked if there is like a free spot in one of the places and we can put our EV samples in there to see if it can pick up better like the differences, because my concern is the overlap between the groups. You know, you won't be able to set up a threshold because you have quite a lot of overlap. The same happened with the cryptic exons, for example, for TDP43. It's very promising, and it will be nice as an MSD assay, but the overlap that they have between the groups, it's very, very large. So yeah.
So you think it could be I mean that like freely circulating TDP43, be it in plasma or in CSF, we know that from many studies that it doesn't really work. So do you think it may be a sensitivity issue, or the trick is really like this recent paper suggests that you have to go to the extracellular?
I think it can be either sensitivity or either specificity, because, you know, we don't know the epitopes of the Simoa assay. They are not going to disclose that. So we really don't know which form of TDP they are picking up. What are we measuring? Because if we are measuring total TDP43, maybe we are not going to be able to pick up differences. But then if we have like a more specific assay, or we kind of point it more to the toxic species or specific species, depending on what, that will be better.
Great. Lots of work to be done.
Yes.
Oh, good. Okay, thank you very much. Yeah, actually, I was thinking about the comparability of your data with sporadic.
Yeah.
But you already mentioned that. But I think, oh, yeah.
No, but yeah, it's true that, you know, we try to extrapolate things, or the way we've been doing the studies is to do a pilot study in a sporadic FTD. If we see something, then we do it in genetic FTD. But I think there is still a big gap on trying to translate from genetic FTD to sporadic, because in sporadic, you really don't know what's going on. You will have co-pathology, probably. You don't know what's
And in
changing.
too. In genetic cases too.
Yes, yeah, definitely.
Yeah.
Mm-hmm. Yeah.
Yeah.
Yeah. Mm.
Yeah, at least that happens in genetic Alzheimer's cases. But okay, I mean that's quite difficult, but very but amazing. But I was just making myself and yourself another question, but this is a sort of a brain banker question. You have this large cohort, beautiful cohort, lots of centers. What is the brain donation rate? You know,
I really don't know. Sorry, because I'm not the coordinator, you know, so I don't know. But so the thing is that it's not that all the cohort. So most of the participants are still alive. And I know that at UCL, we do have quite a lot in the brain bank. And we also have patient lines as well, stem cell lines from patients from GEMFI. But I really don't know which is the rate. What I've seen, I think probably depends on the country, but what I see in the UK that I was very surprised when I moved there is that everyone is very willing to donate, to participate in research studies and everything. So I really don't know in GenFi in general, which is the proportion of donation. Yeah.
Okay, because actually you have this high variability of data, for example, in progranulin, but one thing that some of this variability can be explained when you look at the
Yes.
brain tissue.
Mm.
So, but that's for the future.
Yeah. No, but actually we were thinking with the proteomic study that will be something that it will be very interesting if we can get matched brain from the CSF samples that we've used, and then match plasma and do kind of cross tissue study, which I've seen a couple of them and they are amazing.
Yeah.
But yeah.
And even for the C9 mutations, for the tau mutation, they have a high variability, pathological phenotypic variability.
Yeah.
But well, thank you very much.
Thank you.
Beautiful.
Thank you.
Perhaps a lot of findings, small findings, most of them. But among all of these findings, what do you think that are the most promising finding to guide the future clinical trials, to be extrapolated or examined in the sporadic cases? What do you think that is the most interesting and promising findings?
Well, I'm very, very excited about the lysosomal changes that I saw that were very specific of MAPT, because I know that the therapeutical interventions of progranulin, a lot of companies, they are focusing on endolysosomal targets. So I don't know if that will be actually the best approach looking at this, or maybe which will be the outcome measures or the of that clinical trials, if they are measuring, for example, BMP4 and things like that. I will say that will be very interesting to continue to look into what is going on the endolysosomal system in MAPT, and as well as the synaptic proteins. What is different? There is something different on the synaptic dysfunction between Alzheimer's disease and FTD, even though it's a synaptic dysfunction, but we still don't know what's going on in FTD, or which are the markers for FTD, which proteins are implicated in there. So yeah, hope that answers your question.
So I had a follow-up question to what Alberto asked about heterogeneity, phenotypic pathological heterogeneity in even in mutation carriers. As far as I know, there's only some MAPT mutations produce AD-like tauopathy, like 3R, 4R tauopathy, whereas most do not. And I wonder, since you found these differences of this AD-focused biomarker panel only in these MAPT carriers, do you have an idea? Probably, yeah. Do you have an idea how big the proportion of 3R, 4R
I don't know.
patients is in this?
Yeah, I think we do have that information, but we haven't looked into that. Actually, it will be very interesting to see among our population of MAPT mutation carriers, which are the changes. Also, we don't have any pre-biomarker data on tau or p-tau, because there have been some studies and they don't show any differences in FTD. So yeah, we haven't looked into that in GenFi.
There are some tau PET studies who show like very minor signal increases in MAPT mutation carriers, except for the 3R, 4R.
Okay. It will be very interesting to look into that. Yeah, we do have the information about the mutations, so that's something that we can have a look. Yeah, thank you.
So if there are no additional questions, then I think we can close the session.
Okay.
Thank you again.
Well, thank you very much.
We have 3 minutes.